Hello, good afternoon. I'm Dr. Kalpana Patel from Environmental Health Center, Buffalo, and it's great to be present with you here in Las Vegas virtually. I want to thank this opportunity to thank Dr. Clearfield and his team. And today I'm going to discuss chronic fatigue and various aspects of dyshomeostasis and mitochondrial dysfunction. And please bear with me as I have to switch it to the share slides. I think it is this probably. Yeah. I do not have any financial disclosures as no relevant relationship with the commercial interest to disclose. The main objectives of my talk is what is chronic fatigue, what are the atypical manifestations of chronic fatigue, which I am going to emphasize, understand homeostasis, and how it is related to chronic fatigue, discuss mitochondrial dysfunction, mitochondrial function in health and disease, and mitochondrial dysfunction, how it is with chronic fatigue intertwined. So there are multiple core criteria to diagnose chronic fatigue. It is the impairment of normal ability to function due to fatigue, along with the post-exertional malaise, worsening of illness after physical, mental, and cognitive effort. There can be a poor, unrefreshing sleep, as well as cognitive impairment. It may be associated with POTS, which symptoms of dizziness and orthostatic intolerance, which can be moderate to severe in 50% of the cases, and related diagnosed by the heart rate variability. And most of physicians, those who are present, can do in their offices. And post 6 months of medical investigations and supportive treatment with no change in symptoms are not uncommon. Most patients remain undiagnosed and unable to access expert medical interventions despite evidence of significant debilitation and chronicity as seen many of our patients. So chronic fatigue can be also equal to dyshomeostasis, as chronically ill patients are always in state of constant dyshomeostasis. The atypical manifestations can be increased in total toxic burden, as well as increase in the total body burden, which results in dyshomeostasis. Also, there can be a mitochondrial dysfunction, impaired microcirculation, immune deficiency, immune dysfunction, having allergies, and autoimmune activation. So to maintain the homeostasis is role of ground regulation system. So what is ground regulation system? It is a communication system between external and internal environment required for the maintenance of homeostasis. There is a rapid, dynamic, harmonic, energy efficient automatic response, which is part of the every defense mechanism in our body. It enables body to adapt to changing environment and maintain internal environment within the physiologic range. And that is the main purpose of the local ground regulation system. This system, there is ever-changing dynamic response for obtaining and maintaining optimal physical and mental energy. It is an information network which controls the influx of nutrients and oxygen, as well as efflux of cellular waste, ingressing and outgressing of nerve impulses, maintenance of isotonia and isoosmia, which are part of many patients having a problem with the postural intolerance and tachycardia. Pain, fatigue, and weakness are the early manifestations of the homeodynamic response. The ground regulation system has many connective tissue matrix as shown here, and it is 40% of the body's tissue surrounding every organ, including fascia, tendons, and ligaments. 87% of the dermis and submucosa have connective tissue matrix, which is made up of 3 types of cells like mast cells, fibroblasts, and macrophages. And this amorphous material, which has the proteoglycans, glucosaminoglycans, and hyaluronic acid. This is the collagen fibers, and these are the elastin fibers, and also the capillaries, as well as the somatic sensory fibers, as well as autonomic nerve endings, as shown here, and basement membrane, which interfaces between the organ cells and connective tissue matrix. 85% of the subepithelial mast cells are in contact with the 0.2 micron sensory nerve fibers, as we see here, containing substance P, which constantly triggers neuroimmune endocrine response in many toxic chronic infection patients and release the histamine, serotonin, acetylcholine, norepinephrine, and other kinins, and may result into the mast cell activation and massive release of histamine. It is not an isolated new syndrome, but it is body's response to increase in total body burden, and it does not need drug management if all of the factors of the environmental pollutant load are addressed. So remember, connective tissue matrix is a mesh between the blood vessels, lymphatics, and nerve endings, cell parenchyma of the end organs, communication of the disturbances of tissues, and external and internal environment. It regulates nutrition and its disposal. The basement membrane, as shown here, it is rich in vitamin C and calcium. It quenches free radicals. Nutrients enter through basement membrane, but when pollutant damage it, the organ malfunctions. Pollutant overload stimulates excessive formation of PG-GAGs, and oxidative free radicals trigger inflammation and basement membrane malfunctions. As you see here, this is the extracellular connective tissue matrix, and these are the capillaries and canaliculi, and this is the venous system, and then these are the lymphatic sieves. And PG-GAGs are they slag in this lymphatic system and impair the lymphatic drainage. So connective tissue matrix inflammation occurs, and however, when there is a less caloric intake and fasting, it clears these PG-GAG slags, and impaired nutrition returns to normal. Now, let's look at the macrophages. It is the soul of the immune system, and this is where the immune system starts responding to the incitants. There are 2 different kinds of macrophages, M1, which are called inflammatory macrophages, responding to IL-6, and M2, anti-inflammatory, responding to IL-10. M1 are responsible for inflammation. M2 is responsible for proliferation, remodeling. There are 4 types of macrophages. They support the extracellular matrix deposition, angiogenesis, and also plays role in angiogenesis via VEGF. As you see here, the M1 responds to killing response, and it is noticed here, it produces large amount of reactive oxygen species, cytotoxicity results, and killing occurs, where M2 macrophages are the responsible for cell proliferation, collagen deposition, and has oxidative phosphorylation as their energy production. M1 uses the aerobic glycation and M2 uses the oxidative phosphorylation. Now let's look at the total environmental pollutant load, as it is one of the major factors responsible in causation of dyshomeostasis when it exceeds the total body capacity. So these pollutants can be derived from air, food, and water. These are the non-specific load, and what derives from the air is the outdoor pollution, and outdoor pollution can be sulfur compounds, ozone, carbon monoxide, particulates, EMF, lead, cadmium, mercury, pesticides, and that can be from food and water too. EMF radiation, radon gas, microwaves, as well as the natural gas oil, pesticides, formaldehyde, solvents, fumes, and carpets. The food has man-made chemicals, natural chemicals, as well as the biological pollutants and emotional trauma as described here. For your detailed instructions, you can make a copy of this slide and see what applies to your patient. And so far, the specific environmental load is concerned, it can be Streptococcus pseudomonas or Lyme and co-infection, or mold and mycotoxin, chlorine, or ionizing radiation. So in order to understand the total environment pollutant load and what it does to the body, this sand pile model is a very good way to demonstrate the cumulative impact of environmental stressors on our bodies over time. This is an elegant visual metaphor, and it also demonstrates how we are continuously refreshing ourselves at ever higher levels of resilience. Higher level of resilience and adaptive capacity. So as we see here, the grains of sands being added to gradually evolving sand pile, and occasion the disruption occurs and the repair happens. So grains of sand being added to precipitate the partial collapse of the sand pile, but also by the means of sand pile being repaired, we are able to build itself back up, and each time at new level of homeostasis. And that's what occurs in our body. The system will therefore have been able not only to manage the impact of the stress, but also to benefit from the stress. So thus we have to remember that state of our health depends upon the total body burden derived from the pollutants for the air, food, and water, like inhalants, food chemicals, infectious agents, and electromagnetic field, as well as the inner body pollutants, like a total emotional load, nutrients, and the genetics, polymorphism. So that predisposes one to have more health issues. So how does the body respond to total environmental body load? There are many different mechanisms. The one which is adaptation, bipolarity, spreading, switch, and biochemical individuality. Adaptation is an acute survival mechanism. It is the body's way to get used to acute exposure. However, ongoing exposure results in masking, where the pathologic changes continues to occur, but person may not have any symptoms. But if it continues, then maladaptation develops. One has symptoms and also the pathologic changes. And during this phase, the nutrient depletion occurs. And in phase of total pollutant load, in presence of nutrient depletion, the inflammation occurs and fixed name disease develops. And bipolarity is another way the body responds, where there's a stimulatory effect, and during that one has a stimulation like happens with the alcohol ingestion, and initial phase of stimulation follows with the withdrawal phase. Similarly, it occurs with chocolate, sugar intake, and many other the inhalants like chemicals, which a sniff gives the stimulatory effect. And that's why the glue sniffing is not uncommon in children. The spreading mechanism is another way of responding, where one develops the symptoms in respiratory symptoms, ENT symptoms, and goes into the brain and develops the cognitive dysfunction, develops the abdominal pain, or increase in urinary frequency. There is another phenomena which is called switch response, and that the reaction changes from one system to another system. So one may have allergic rhinitis or asthma at one point, and then they disappear and develop the symptoms switches to the another organ like a GI system. And biochemical individuality is a very important aspect, how the body reacts to the pollutant load. Each individual is biochemically different, having a different type of immune detox system, vitamin, mineral, antioxidant system, protein and carbohydrate and fat, nutrients. So specific environmental load also is very important for one to develop the dyshomeostasis and how one develops homeostasis to dyshomeostasis. When total body pollutant burden increases in the connective tissue matrix and not able to contain into the local regional symptoms, one develops the secondary amplification and symptoms in a neuroimmune endocrine system, including cardiovascular system, as well as the CNS activation occurs in reticular formation, hypothalamus, limbic system, area postrema of fourth vertical and pineal gland. And all of these are the rest of the body's alarm reaction through the autonomic nervous system and adrenal gland. So now, when there is excess total body burden in presence of nutrient deficiency, one develops the inflammation, which may develop as a hypersensitivity. And initially, one may have a physiologic response where the incitant has a stimulatory response, then it comes down to the normal level. But if there is a constant barrage, then, and when masking occurs, when there is a delayed recovery, and when there is maladaptation, you can see the resonance deterioration response. So, and it can be cold or it can be warm, and one may manifest as angioedema or chronic vasculitis, fibrosis, anaphylaxis. If this response becomes the aperiodic response and does not have any stimulatory phase, but just constant downward domino effect, and these resonant degeneration, then one develops the metabolic deterioration, enzyme deficiency, and development of diabetes, spondylosis, vascular diseases, scleroderma, arteriosclerosis. So there, and then inflammation can manifest as a combination of hypersensitivity and chronic degenerative disease, and which may develop as a degenerative disc disease, myofascial hypersensitivity, which we see as fibromyalgia and many of our chronic fatigue patients, and degenerative joint disease is not uncommon. So now let's look at the chronic fatigue and mitochondrial dysfunction. chronic fatigue is equal to mitochondrial dysfunction? Yes. So let's look at mitochondria. So mitochondria are dynamic double membrane bound organelle present in the cytoplasm of every cell of the human body except that of the red blood cells and skin. Most cells have about 2,500 mitochondria, making up 1/5 of the cell volume. However, germ cells have 100,000 and platelets have 2 to 5 mitochondria. The mitochondria are semi-autonomous. They possess their own genome and machinery for replication, transcription, and protein synthesis. And mitochondria has about 15% of the total cellular glutathione pool and 30% of the SM, S-adenosylmethionine for methylation. Now let's look at the structure of mitochondria. Mitochondria is a double membrane. So this is their outer membrane, and this is the inner membrane. And there is the intermembrane space that you can just see here. And this is the inner membrane. And these are the crusty, and this blue color is their matrix. And this green color is the mitochondrial chain. This is the inner membrane. This inner membrane is where there's electron transport change and where the ATP complexes occur. However, in matrix is the place where the cytochrome C oxidase is present throughout the matrix except near the mitochondrial DNA. And mitochondrial matrix also has a metabolic pathways. A THC cycle with oxidation and heme synthesis cycle occurs in mitochondrial matrix. So now let's look at the mitochondria in a young and old. In a young, there are about there are multiple mitochondria producing many molecules at ATP and only 2 molecules of ROS, whereas in an old, aged, and chronically ill patient, they have only very few mitochondria producing only 2 ATP and 6 ROS. And look at the size of this mitochondria. They are unhealthy, large mitochondria, and they are likely to be poorly phagocytized, leading to further damage and enlargement of the cell, ready to be phagocytized. Damaged mitochondria and lipofuscin inclusions during chronic persistent Lyme and chronic infection occurs very commonly. Now, oxidative phosphorylation is the very essential part for the ATP formation. So it occurs in a matrix of mitochondria. Mitochondria generates ATP by using oxygen as a final electron acceptor. There are 2 main steps for oxidative phosphorylation: electron transfer and chemoosmosis. Both of these processes use the moment of the charged particles to transfer energy. Glycolysis only produces 2 ATPs, while oxidative phosphorylation produces 32 ATPs, while electrons are shuttled through the electron transfer chain by using oxygen as an electron acceptor. Now, here see that enzymes involved in the electron oxidative phosphorylation, that electrons move from position 1, 2, 3, 4 to 5, and eventually finally it is transferred to electron acceptor oxygen molecule, and ATP is released with the help of ATP synthase. So it is important to know that NADH is the beginning molecule, converted to NAD, and then mixed with the ubiquinone or cytochrome C, which is organic molecule, and it moves freely within the inner membrane of the mitochondria. So what are the functions of this mitochondria besides ATP generation? Well, it is important for the heat production, calcium homeostasis, regulation of membrane potential, formation of steroids, testosterone, estrogen, and hormones, detoxification of ammonia and liver, apoptosis, programmed cell death. High caloric intake produces the dysfunction and increase in apoptosis. However, caloric restriction increases the mitochondrial efficiency. It's very important to understand that 4% of oxygen we breathe is used for the mitochondrial oxidative phosphorylation, where with 90% of the free radicals are generated by this process of oxidation in mitochondria. So there is a big price to be paid for every molecule of ATP generation. Now there is another modality by which the mitochondrial function can be improved, which is photobiomodulation. The monochromatic LED laser light 660 to 810 frequency promotes the normalization of the electron transport chain function in mitochondria. The mitochondria are principal photoreceptor cell of the cell which accepts light, and activated by the light, it increases the ATP production, and it modulates the chronic stress, promotes normalization of the dysfunctional state, and upgrades normal function by having direct effect on cytochrome C oxidase system. Several transcription factors are also upregulated by a low-level laser beam therapy, a photobiomodulation, and it modulates the reactive oxygen species. So now let's look at the interrelationship of mitochondria, NAD, and sirtuins. So Harvard study described close association between the sirtuin gene, mitochondria, and NAD. Sirtuin genes are found in every cell cytoplasm and nucleus in mitochondria. SIRT1 gene responsible for critical biologic functions like DNA expression and reduced aging process. SIRT1 gene induces formation of new mitochondria only in the presence of NAD. However, NAD controls the health, number, and functions of the mitochondria and has a downstream effect on the most of the cellular functions. So NAD activates SIRT3 and keeps the mitochondria running smoothly, essential element of cellular metabolism and DNA damage repair. However, reduced levels of NAD reduces the sirtuin activities, reduces the communication between the cell nucleus and mitochondria at cellular level, and adipose tissue at a systemic level, dysfunction in hypothalamus, and that results in the dysfunction of endocrine and nervous system. Hypothalamus plays a big role in many essential functions of the body, and when it dysfunctions, it causes the dysfunction in body temperature, thirst, appetite, weight, emotions, sleep cycles, and a host of other functions. So many symptoms develop in patients with chronic fatigue syndrome. So chronic fatigue has an adverse effect on central nervous system, which is induced by the reactive oxygen species. As the central nervous system has a mitochondria that have a little resilience to minor illness and causes the minimal trauma that triggers sudden, massive, irreversible damage. The body, 20% of the body's oxygen consumption is done by the central nervous system, and having the high levels of polyunsaturated fatty acids, it is susceptible to free radical damage and results in the oxidative damage, as central nervous system is relatively deficient in oxidative defenses, as has a poor catalase activity and moderate SOD and GPX activity. Some regions of central nervous system has a higher content of iron and ascorbate. Thus, through the Fenton-Haber-Weiss reaction, it generates the more reactive oxygen species. So chronic Lyme patients first develop cognitive dysfunction and then progressive loss of cognitive function. If ongoing damage is not controlled, may develop neurodegenerative disorder and dementia. Environmental toxins, viral infections, and bacterial infections and their toxins generate ROS molecule and RNS molecule through the no-ono cycle, which result in oxidative stress and free radical damage. Major ROS are byproducts of aerobic metabolism. They are superoxide anions, hydrogen peroxide, hydroxyl free radical, and peroxynitrite, which are very destructive molecules, damaging many cell membranes and inner membranes. ROS also cause serious damage to enzyme complexes in lipid-containing inner mitomatrix, where oxidative phosphorylation occurs and outer membranes of mitochondria are also damaged. Oxidation leads to loss of their biological properties and eventually to cell death. Mitochondrial reactive oxygen species act as a signal messengers and modify operation of many enzymes in different cell compartments. And mitochondrial ROS are important in execution of programmed cell death. Thus, there are many functions of mitochondria. To summarize: ATP production, signaling molecule directing different cells to do different tasks, neurogenesis signaling, stress signaling, inducing the overproduction of osteoclast, resulting in osteoporosis, commonly seen in chronic Lyme patients for a long duration. So during the progression of chronic fatigue in chronic Lyme or chronic infection, chemical sensitive, and other chronic diseases, mitochondria goes through profound alteration in function that lead to reduced generation of ATP and chronic fatigue, enhanced production of reactive oxygen species and nitrogen species, and disturbances in nitroredox, loss of calcium, buffering capacity, which initiate cascade of deleterious events within the cell, and damaged mitochondria release several pro-apoptotic proteins that translocate into the nucleus to induce DNA fragmentation and cell death. This alteration in mitochondria contribute to cell degeneration and death. And so it is very important to remember that mitochondrial dysfunction is equal to chronic fatigue. Mitochondrial dysfunction, when proceeds, it results in the damaged mitochondria. Damaged mitochondria will result into DMA, damage, and cell death. So chronically ill, chronic fatigue patient just go in a downward domino effect and becomes bedridden and have no life. So mitochondria also has antioxidant system, first line and second line system. First line defense has an enzymatic antioxidant system and which directly react with the ROS and decrease in ROS production, such as a SOD1, SOD2, and glutathione peroxide. And when it is low, the neuroinflammation occurs in the brain. Second line defense is non-enzymatic antioxidant system, which remove ROS and other oxygen derivatives. And the important nutrients are vitamin A, C, E, polyphenols, glutathione, and other antioxidants. Vitamin E plays an important role in cellular defense against lipid peroxidation and cell membrane by free radicals. Now let's look at the glutathione. Glutathione is the most abundant thiols in cell. It does not mitochondria does not synthesize glutathione. It is transported from the cytoplasm to mitochondria by the transporter, alpha-ketoglutarate. Mitochondria glutathione pool amounts to approximately 15% of the total cellular glutathione. Mitochondria glutathione pool plays a critical role in cell protection. Vitamin D acts as a catalyst for glutathione production. It is a primary cellular redox environment agent. It is antioxidant and also antitoxin, which augment detoxification of xenobiotics and biotoxins. Not only that, but it's also free radical scavenger. It is intimately related to the regulation of nitric oxide cycle. Thus, it is the nitro redox agent and important in increasing the stem cell output from the bone marrow, DNA synthesis and repair. Thus, nitric oxide is also very important in protein synthesis, prostaglandin synthesis, amino acid transport, enzyme activation. Every system in the body can be affected by the state of glutathione system alteration, especially the immune system, nervous system, and GI system, as well as lungs. Overproduction of reactive oxygen species occurs. Oxidation of the glutathione results in mitochondrial dysfunction and disease. So now let's look at the microcirculation and chronic fatigue. About 75% of the body's blood circulation occurs in the microcirculation. Therefore, it is a very important system to remember that it is an important contributor to the development of the disease if it is not functioning normally. It consists of arterioles, venules, capillaries, as well as the precapillary sphincter. It plays a critical role in regulating tissue perfusion and also is a parakine modulator of the local tissue environment. There are certain facts of the microcirculation which are not commonly known, like blood does not flow continuously through capillaries in this microcirculation. Flow is intermittent. It is turning on and off every few seconds and minutes, like pulse is intermittent flow is physiologic and it enhances oxygen and nutrient extraction. Its communication appears to flow constantly, unimpaired or augmented by this phenomena. Exposure to toxins and toxic substances such as the biotoxins, mycotoxins, lead, mercury, hydrocarbons, and pesticides disturbs microcirculation. It alters the capillary sphincter mechanism. Shunting of the blood from arterioles to venules occur, but it escapes the capillary perfusion, which results in a high levels of venous oxygen concentration. Cellular oxygen concentration becomes minimal, and venous oxygen concentration increases, which results in the higher concentration of oxygen, PVO2. So when you do venous gases, and if you find a high PVO2, then you know patient is suffering from the issues of microcirculation problem. Local tissue hypoxia results, local acidosis induces release of free metals like copper and iron from the transporting proteins. Free metals promotes reactive oxygen synthesis production and activate the signal transduction. This results in altered mitochondrial oxygen diffusion, inefficient oxidative phosphorylation, impaired ATP generation. Inability to extract sufficient oxygen results in inefficient detoxification. Impaired detoxification occurs in all pathways. The take-home message from this is that even though mitochondria is normal, if microcirculation is abnormal, then there will be ineffective oxidative phosphorylation and impaired ATP generation and poor energy. So in this situation, if you provide the oxygen therapy, mitochondria is normal, so can always produce more ATP and patient starts feeling better. So this is in a chronically ill patient, one should always do the venous blood gases to see the state of microcirculation. And then another fact is impaired oxidation by cytochrome C oxidase system also results in low ATP generation and fatigue results. So when you give them photobiomodulation treatment, they start feeling better, then you know where the problem is. Now let's look at the chronic fatigue and immune dysfunction. We know that immune system is forever evolving through the functional plasticity of the immune system. Let's say that the CD4, CD8, Th1, Th2, Th17, Th23 are very important phenotypes playing a great role in the maintenance of health along with the other systems. But if we see that these are the T cells which at birth, which are released from the bone marrow, then it goes to the thymus gland and get their kindergarten education. They go to the lymph nodes, they go to the elementary education. Then they go to the target organs like skin, lungs, and intestine. They go through the high school education. And in the university education, these cells become the CD4, and then T cells become CD4, and then they become Th1, Treg, Th17, T20, Th23, and Th2. So they play a great role. There are two parts to the immune system. One is the humoral immune system, one is the cell-mediated immunity, and then there are secretory IgA, immunoglobulin A, G, M, and IgE. IgG subclasses, complement cascade, immune complexes, and cytokine level can be measured, and it gives a great reference number in the investigation of the chronic fatigue. And cell-mediated immunity is also should be looked at. T cells, B cells, helper cells, suppressor cells, natural killer cells, also their phenotype and cytokine profile. Now, when you are evaluating and going through all the results, you may find some patients have a high CD4, CD8 ratio. What does that mean? It is a marker of immune activation, and it is detected in a patient even with the heavy metal exposure. While we see the low CD4, CD8 marker in immune deficiency patients, those who have exposure to pesticides, xenobiotics, biotoxins, and high natural killer cells is seen in viral infection as well as early stages of cancer. We have seen the patients also having the autoimmune activation, high CD4, CD8 ratio. So this is very important to do the phenotype. The lymphocyte flow cytometry will give you these results. Chronic Lyme disease and chronic mycoplasma infection, bacteria persist as they Borrelia burgdorferi evade and suppress the immune system. And then adaptive immunity becomes poor and results in the persistent infection, persistent inflammation, cytokine also our profile gets altered, and poor adaptive response emerges, and autoimmune activation also occurs. There's a IgM is the first antibody secreted during the immune response, controlling bacteremia and activating the classical complement pathway. And then after a few weeks, it switches to IgG, soon after the infection. So there are 4 types of IgG, and if one is deficient in one IgG subclass, then one may not have the optimal secondary IgG response. Serum IgG levels remain high throughout the infection in cases of Lyme disease and chronic mycoplasma infection, which suggests also that there is a chronic persistent infection. Strong ongoing production of IgM is predominant only in chronically ill patients who are immunochallenged. So the inflammation, we have looked at it before, but remember, dynamic nutritional status is essential for the proper homeodynamic response in the presence of excess total body pollutant load. Prevention, reversibility, and control of the disease are dependent on the state of nutrition. So if you have optimal nutrient level, even in the presence of excessive total body pollutant load, one may not develop inflammation, and that is the take-home message. But sometimes the level of nutrient may be normal in the blood, but maybe organ-specific deficiency may be there, and these are the cases where one develops the inflammation in organs like neuroinflammation or liver inflammation, cardiac inflammation. So one has to be aware that what you measure in the blood is not adequate. Chronic infection triggers the oxidative stress, and oxidative stress induced neuroinflammation, which is the hallmark of neurodegenerative diseases. So here you see the burning fire, which is the fire in the limbic system and hypothalamus pituitary axis. So this slide is for you to print and use in your practice, that what you do when you see a chronic fatigue neuroinflammation patient, what is the etiology. And then this slide is again for you to print and see how you can intervene the mitochondrial dysfunction and oxidative stress in excess total body load. This is the methylation pathway, that when there is an increase in total body load, there is a problem with the methylation pathways, and it results in the low glutathione levels, neurotransmitter abnormalities, the sleep onset disorder, melatonin deficiency, and metabolic abnormalities, and problems with the redox pathways. Then again, dysfunction in mitochondria is produced from reactive oxygen species, and extensive oxidative damage occurs, immune activation follows, increase of the circulating IL-6 indicates the immune activation, changes in the innate and adaptive immunity, release of the pro-inflammatory cytokines and signaling molecules like NF-kappa B and TNF-alpha occurs. Oxidative stress also depresses vitamin D, but also vitamin D receptor expression is depressed. Mitochondrial vitamin D hydroxylase activities are reduced, and glutathione synthesis is compromised. Thus, low vitamin D and altered receptor functions are linked to risk of autoimmune disease. Thus, in summary, evaluation of sick patients must be done prior to providing optimal treatment plan. Do not have a treatment plan before you check the patient for their homeostatic dysfunction, mitochondrial dysfunction, vitamin D level, glutathione level, NAD level, and other contributing factors like what is there to increase their total body pollutant load. Every part of the homeostasis and ground regulation system dysfunction must be considered. Role of dysfunctioning connective tissue matrix cells, the mast cells, which are the most important cells, and it plays a great role. Macrophages are the soul of the immune system, and the fibroblasts are also important for the formation of PGGAGs. They are also important for the formation of the elastic fibers, connective tissue matrix. And when there is a unequal function of fibroblast and macrophage, where then the problem with the fibrosis resolves, because the macrophages are not completely removing the excessive production of PGGAGs. So this is very important, because if you do not have enough amount of PGGAGs, your body system is not going to function well. Vitamin C plays a great role in maintenance of the basement membrane and function, and it protects the basement membrane from the oxidative damage and organ dysfunction. So vitamin C replacement can be very helpful, and optimal vitamin C function can be maintained by supplementing high dose of vitamin C, but it alone does not work. It has to be supplemented with the other nutrients. Remember the total body burden. Evaluate the total body burden and activation of the neuroimmune endocrine axis. Why there is a secondary system amplification of the neuroimmune system occurs must be completely checked for. The role of immune dysfunction and autoimmune dysfunction is important in a chronic fatigue, and look for the causation. What is causing the immune dysfunction? What is causing the autoimmune activation? Autoimmune activation is just the beginning of the autoimmune disease, but you know it can be reversed, but one does not have to really suffer from it. So it is important to work and try to treat the cause. If you have a chronic infection, if you treat chronic infection, you will see the reversal of the autoimmune activation. Then also pay a great attention to the role of mitochondrial dysfunction and electron transport chain, role of the NAD in maintenance of the sirtuin genes. If the patient does not respond to your initial treatment, then you may give a patient NAD, intravenous infusion. However, we have noticed that if you have not addressed the primary problem, patient will get worse with the NAD infusion. So it is important that what stage of the disease the patient is. You first try to lower the total body pollutant load. You try to correct the nutrient deficiency and bring the patient at different level of homeostasis, as we were showing in our sand pile model. And then you give them the NAD. It may do a great deal for the patient. But before you give NAD, you may want to try the glutathione and see that by increasing the glutathione pool, how much difference you can make in the patient reversing the chronic fatigue symptoms. And role of the glutathione, remember the powerful antioxidant and antitoxin and good nitro-redox agent. Role of PBM should not be forgotten, because every patient should be given a chance of this photobiomodulation therapy, as it does have a great resolution of symptoms. Particularly, we have noticed that the activation of mitochondria and musculoskeletal system occurs, but cognitive dysfunction is greatly improved. Methylation also should be supported, and first find out patient has any polymorphism and any methylation enzymes and pathways, and then address accordingly. Role of vitamin D cannot be underestimated. Vitamin C should be equally important, and other antioxidant enzyme system should be supported. And microcirculation can be supported by giving the oxygen treatment. But first, do not forget to do the venous blood gases. And when do the venous blood gases, make sure that no tourniquet is applied and blood is drawn. Measurement of the venous oxygen is done, because that's the one molecule that you are very interested in. And if it is high, then you can give patient 18 days oxygen treatment. You can give about 6 liters per minute, and you can give for 2 hours. It has to be a continuous flow through the special apparatus. And hyperbaric oxygen has been found very helpful. Also, the oxygen ozone treatment can be very helpful in these individuals. So it is very comprehensive evaluation must be done before you start the treatment, because there are many facets to the treatment, many prongs to the treatment, and you can have a gradual approach. Do not try to treat with everything the super sick patients, because you may make them sicker as their metabolic pathways are not open completely, and they are in imbalance. And if they are in imbalance, you have to go low, slow, and that will be very helpful. Thank you so much for your participation in this lecture, and thank you.